You begin at dawn. The air is cool, almost silver in its stillness, and the world feels as though it is holding its breath. Before you, a single leaf waits. It does not move much. It does not ask for anything. And yet, it is ready. The first light arrives quietly. Photons stream from the sun, having traveled nearly 150 million kilometers through space. They have crossed emptiness for 8 long minutes just to arrive here, to this small green surface. When they reach the leaf, they do not knock. They simply touch. You might imagine light as warmth, or brightness, or color. But here, at the surface of the leaf, light is something even smaller. It is a packet of energy. A tiny delivery of motion. Each photon carries a specific amount of energy depending on its wavelength. Blue light holds more energy than red. Infrared carries less. The leaf does not see color the way you do. It feels energy. The outermost layer of the leaf is called the epidermis. It is thin and mostly transparent, like a clear window pane. Its role is protection, not absorption. It allows light to pass inward, filtering only slightly. Already, you sense a boundary. Not all light is welcomed equally. Some reflects away. Some passes straight through. Only certain wavelengths will be useful deeper inside. You move inward. Beneath the surface lies a layer filled with cells shaped like elongated columns. Inside these cells are structures called chloroplasts. And inside those chloroplasts is chlorophyll, the pigment that makes the leaf green. Chlorophyll is often described in textbooks as a molecule that absorbs light. But here, in this quiet morning, it feels more like a listener. When a photon strikes chlorophyll, something subtle happens. The energy of the photon excites an electron within the chlorophyll molecule. The electron does not leave immediately. It simply rises to a higher energy state, like someone briefly standing taller in surprise. This moment is the beginning of photosynthesis. To capture light does not mean to grab it and hold it still. It means to convert its energy into a form that can be passed along. The excited electron transfers its energy to nearby molecules in a carefully arranged network. This network is known as a photosystem. You might picture it as a circle of friends passing along a message, each one positioned at just the right distance. But here is something gentle and important: not every photon that touches the leaf is captured. Some bounce away. That is why leaves look green. Chlorophyll absorbs red and blue wavelengths most strongly, but reflects green. What you see as color is actually rejection, light that was not taken in. Even in abundance, there is selectivity. And even among the photons that are absorbed, not all are used efficiently. If light arrives too intensely, the system can become overwhelmed. If photons strike faster than the electrons can be passed along, energy has nowhere to go. It must be safely released, often as heat. The leaf protects itself from overload. You begin to understand that the leaf is not trying to capture all light. It is trying to manage it. The process continues deeper within the chloroplast. The excited electron moves through a chain of proteins embedded in delicate membranes called thylakoids. As it travels, its energy is gradually transferred, step by step. With each transfer, small packets of energy are used to pump protons across the membrane, building a gradient, a difference in concentration. This gradient is like water held behind a dam. It stores potential energy. Later, that stored energy will drive the creation of ATP, a molecule that acts like a rechargeable battery for the cell. Another molecule, NADPH, will also be formed, carrying energized electrons onward. These molecules will eventually help turn carbon dioxide into sugar. But for now, you remain at the surface, at the first touch. Light arrives continuously. The sun does not pause to ask how much is needed. At noon on a bright day, more than 1,000 watts of solar energy can fall on each square meter of Earth's surface. It seems generous, endless. Yet a typical leaf converts only a small fraction of that energy into stored chemical energy, often less than 5% under natural conditions. Why so little? Part of the answer lies here, in the first encounter. Only certain wavelengths are absorbed. Only certain photons excite electrons effectively. Only so many reaction centers, specialized chlorophyll pairs, exist within each photosystem. When all are busy, new energy must wait or be dissipated. The leaf is not built for maximum capture. It is built for stability. You notice the stomata, tiny pores on the leaf's underside. Though they are not directly involved in capturing light, they remind you that photosynthesis is not just about sunlight. It is about balance between light, carbon dioxide, water, and temperature. Even at this early stage, limits are intertwined. As morning grows brighter, the leaf adjusts. Chloroplasts can shift position within cells, moving to optimize light absorption when light is dim, or to reduce exposure when light is intense. It is a subtle choreography. The plant does not chase the sun. It negotiates with it. And still, photons continue to arrive. Some will begin the chain of energy transfer. Some will be reflected as green. Some will warm the surface. Some will pass through entirely, reaching the ground below. Capture is never total. You feel a quiet truth unfolding: abundance does not erase limitation. In fact, abundance reveals it. The more light there is, the more carefully it must be handled. The leaf's surface is not a greedy collector. It is a careful gatekeeper. Here, at this boundary between sky and cell, the story of photosynthesis begins not with excess, but with selection. The dawn has now turned to full morning. The leaf glows softly, not because it holds all the light, but because it holds just enough. Energy flows inward in measured streams. Electrons rise and fall. Gradients build like silent reservoirs. You remain with that first touch, the photon meeting chlorophyll, the electron lifting briefly in response. And you begin to wonder what lies beyond the surface encounter. If the leaf is selective at its boundary, what deeper structures shape what happens next? You move closer now, beyond the bright surface of the leaf. The morning light still falls gently from above, but your attention slips inward, through the transparent epidermis and into the soft interior. What seemed flat and simple from the outside reveals itself as layered and alive. The leaf is not a sheet. It is a landscape. Inside, cells are arranged like small rooms in a quiet building. Each one holds its own delicate machinery. The air spaces between them allow carbon dioxide to drift inward. The walls are thin but firm, made of cellulose, strong enough to hold shape, gentle enough to allow exchange. And within many of these cells, you notice small green bodies suspended in fluid. They are chloroplasts. Each chloroplast is no larger than a few micrometers across. You could fit dozens across the width of a human hair. And yet within each one, light is transformed into the chemistry of life. You imagine the chloroplast as a lantern floating in a clear pool called the cytoplasm. It has its own outer membrane, and just inside that, another membrane, 2 layers like a careful envelope protecting something precious. These membranes regulate what enters and what leaves. Not everything is permitted inside. Even here, boundaries remain. Within the chloroplast lies a fluid called the stroma. It is calm, enzyme-rich, and gently active. This is where carbon dioxide will later be turned into sugar. But for now, your attention is drawn to the inner architecture, the thylakoid membranes. They are folded inward again and again, forming stacks that resemble neat piles of coins. These stacks are called grana. Thin membrane bridges connect one stack to another, creating a continuous internal network. Why so many folds? Because light reactions happen along these membranes. The proteins that absorb photons and pass electrons, the photosystems, are embedded here. By folding the membrane into stacks, the chloroplast increases its surface area dramatically, without increasing its overall size. It is like building many shelves within a small room to hold more books. The design allows more reactions to occur at once. More chlorophyll molecules. More electron transport chains. More opportunity to convert light into usable energy. And yet, even this expansion has limits. The number of photosystems that can fit within a thylakoid membrane is finite. The spacing between them must be precise. Too crowded, and they interfere with one another. Too sparse, and efficiency drops. Structure demands balance. You begin to see that the architecture of the chloroplast shapes what is possible. Light capture is not only about the sun's generosity. It is about how much machinery is available to receive it. Each photosystem contains a reaction center, a special pair of chlorophyll molecules positioned perfectly to initiate electron transfer. Surrounding them are antenna pigments, arranged to funnel energy inward. When light strikes one pigment, the energy is passed like a whisper through the cluster until it reaches the reaction center. But each reaction center can only process one excitation at a time. If photons arrive faster than electrons can move down the chain, the system must pause. Excess energy cannot be stacked infinitely. It must be released safely, often as heat. The folds of the membrane provide space for reactions, but not infinite space. Even design cannot escape limitation. You notice how carefully arranged everything is. Proteins are not scattered randomly. They are positioned with nanometer precision. The distance between molecules determines how efficiently energy can hop from one to another. Too far apart, and energy is lost. Too close, and collisions disrupt the flow. Within the thylakoid membrane, protons are pumped from the stroma into the interior space of each disk. This creates a concentration difference, a gradient. The interior becomes more acidic. The exterior less so. The folded stacks help contain this gradient. Like narrow reservoirs, they allow protons to accumulate quickly. When they later flow back through a protein called ATP synthase, energy is captured to form ATP. The shape of the membrane makes this possible. You begin to appreciate that architecture is not decoration. It is function. The folds are not aesthetic. They are necessary. Still, the chloroplast is not static. It adjusts. In low light, grana stacks can become more tightly oppressed, optimizing energy transfer. In high light, the arrangement can loosen slightly, redistributing photosystems to prevent overload. Structure responds to environment. And yet, no rearrangement can make the chloroplast infinitely productive. There is only so much membrane. Only so many proteins. Only so much room within a cell. Beyond the thylakoids, in the stroma, enzymes wait for the products of the light reactions ATP and NADPH. These enzymes will carry out the Calvin cycle, fixing carbon into sugar. But their activity also depends on concentration, temperature, and availability of carbon dioxide. The chloroplast contains its own small genome, remnants of an ancient symbiosis. Long ago, billions of years in the past, a free-living photosynthetic bacterium was engulfed by another cell. Instead of being digested, it stayed. Over time, it became the chloroplast. This history explains its double membrane. It once had its own independent life. Now it serves the plant cell. Evolution shaped this architecture slowly. Not for perfection, but for survival. The chloroplast works well enough. It captures enough light. It builds enough sugar. It does not aim for maximum theoretical efficiency. It aims for resilience. You imagine thousands of chloroplasts inside a single leaf, each one quietly performing the same choreography. Photons arrive. Electrons lift. Protons accumulate. ATP spins into existence. All within membranes folded like careful origami. And still, there is constraint. The thickness of the leaf limits how deeply light penetrates. Upper chloroplasts receive more photons than those below. Some cells live in partial shade, even within the same leaf. Architecture distributes opportunity unevenly. You sense how space itself becomes a factor. Light does not reach all equally. Structure determines access. The green cell is not a simple bag of pigment. It is an intricately organized system, layered and folded to manage energy with care. Its design enhances capture, but also defines its ceiling. Even with perfect architecture, even with thousands of chloroplasts arranged like lanterns in a luminous city, everything still depends on the arrival of photons. How quickly they come. How intensely they strike. How their energy is spaced across time. You feel your attention turning outward again, beyond membranes and folds, back toward the sun itself. Because structure prepares the cell for light. But the speed and character of that light, its relentless, racing journey across space, may hold another kind of limit altogether. You lift your gaze beyond the leaf now, beyond the folded membranes and glowing chloroplasts, and you travel outward. Far beyond the blue of the sky. Far beyond the drifting clouds. All the way to the surface of the sun. Here, in a roiling ocean of plasma, light is born. Inside the sun's core, hydrogen atoms fuse into helium under immense pressure. In that fusion, a small amount of mass transforms into energy. That energy begins as high-energy gamma radiation, colliding again and again with particles in the dense solar interior. It can take thousands, even hundreds of thousands of years for that energy to wander outward to the sun's surface. But once it escapes into space, its journey becomes swift and direct. A photon leaves the sun and crosses the vacuum at 300,000 kilometers per second. It does not slow. It does not tire. In just over 8 minutes, it reaches Earth. 8 minutes. After traveling through such vastness, it arrives at something as small as a leaf. You imagine this photon approaching the atmosphere. Some light scatters against air molecules, that is why the sky glows blue. Some is absorbed by ozone or water vapor. But much of it passes through and continues downward. When it finally reaches the leaf, its journey through emptiness ends. It meets matter. And here, something remarkable must happen. The photon cannot simply remain light. It must become chemistry. When it strikes chlorophyll, its energy excites an electron almost instantly, in less than a trillionth of a second. The speed of this transfer is breathtaking. Light moves quickly, and the initial reaction inside the chloroplast happens almost as quickly. But then, everything must slow down. The excited electron enters the electron transport chain. It passes from one protein complex to another. Each step is controlled. Each transfer is measured. Energy is released in small portions, like coins being placed carefully into a jar rather than dumped all at once. Life cannot use energy at the speed of light. If all the energy of a photon were released at once in an uncontrolled way, it could damage the cell. Bonds could break. Molecules could fragment. Instead, the system evolved to stretch that instant into a sequence. You begin to feel the contrast. The photon's journey across space is wild and unrestrained. But inside the leaf, everything is moderated. The chloroplast acts like a patient interpreter, translating the language of light into the slower dialect of chemistry. And here, you encounter another boundary. Sunlight does not arrive evenly. In early morning, photons trickle gently onto the leaf. The photosystems have time to process each one. Electrons move smoothly. ATP and NADPH form at a steady pace. But as the sun rises higher, the flow increases. At noon, photons rain down in torrents. Reaction centers fill quickly. Electrons crowd the system. There is a limit to how many photons can be processed at once. Each photosystem can only handle one excitation at a time. When all reaction centers are occupied, incoming energy has nowhere to go. If not managed, it can generate reactive oxygen species, highly energetic forms of oxygen that can damage proteins and membranes. So the plant slows the system deliberately. It dissipates excess energy as heat through a process called non-photochemical quenching. Pigment molecules shift slightly, altering how energy flows. It is like opening a pressure valve. Too much light overwhelms. Yet too little light creates another problem. On a cloudy day, or beneath a dense canopy, photon arrival drops dramatically. The photosystems sit idle, waiting. ATP production slows. Carbon fixation falters. Growth becomes minimal. There is a minimum threshold of light intensity required for photosynthesis to exceed respiration. Below that point, the plant consumes as much energy as it produces. You realize that photosynthesis exists within a window, not too dim, not too bright. This window varies by species. Shade-tolerant plants have chloroplasts arranged to capture faint light efficiently. Their leaves are often broader and thinner, maximizing surface area. Sun-loving plants, by contrast, are built to endure intense brightness without damage. Still, even the most sun-adapted leaf has an upper ceiling. Above a certain intensity, increasing light does not increase photosynthesis. The curve plateaus. This is called light saturation. The machinery is working at full capacity. Additional photons simply add stress. You imagine a small factory receiving deliveries. If trucks arrive steadily, workers unload them smoothly. If too many trucks arrive at once, the loading dock becomes congested. Packages pile up. Some must be turned away. The leaf is much the same. And there is another subtle constraint, the duration of light. Day and night cycle rhythmically. During darkness, the light reactions stop. The Calvin cycle slows. Starch reserves are used carefully to sustain metabolism until dawn returns. Photosynthesis is not continuous. It pulses with the rotation of Earth. Even latitude plays a role. Near the equator, day length changes little through the year. Closer to the poles, summer days stretch long, while winter brings extended darkness. The speed and duration of light exposure shift with geography. You begin to see how deeply light shapes life. The photon itself moves with perfect constancy, always the same speed in vacuum. But once it touches life, it must be negotiated. Slowed. Distributed. Balanced. The leaf cannot chase the sun. It can only respond. And so, within each chloroplast, within each thylakoid membrane, energy flows at a moderated pace. Electrons rise and fall. Protons accumulate and return. ATP forms and is consumed. The speed of sunlight is infinite compared to the tempo of metabolism. Life survives not by matching that speed, but by tempering it. Because if sunlight continues to increase, if the flow of photons becomes relentless, even careful moderation may not be enough. And you begin to wonder what happens when brightness becomes burden, when abundance presses too hard against the limits of structure and speed. You stand now beneath a sky without clouds. The sun is high. The air shimmers slightly. Light no longer arrives in gentle threads, it pours. What felt nourishing at dawn now feels relentless. Photons descend in dense waves, striking the leaf again and again. The chloroplasts inside are already working, their reaction centers turning over rapidly, electrons passing down chains with practiced precision. But there is a limit to how fast this machinery can move. Each photosystem requires a brief moment to reset after transferring an electron. The reaction center chlorophyll pair must return to its original state before it can accept another photon's energy. This reset happens quickly, in fractions of a second, yet under intense sunlight, photons arrive even faster. You begin to sense congestion. When photons hit faster than electrons can move forward, excited states accumulate. Energy lingers where it should not. And lingering energy is dangerous. Inside the chloroplast, oxygen is abundant. It was produced here only moments ago during the splitting of water molecules. Normally, oxygen drifts away harmlessly. But in high-energy conditions, oxygen can react with unstable electrons to form reactive oxygen species, molecules such as superoxide and singlet oxygen. These are not gentle participants in metabolism. They are highly reactive, capable of damaging proteins, lipids, and even DNA. They can alter the very membranes that hold the photosystems in place. Left unchecked, this oxidative stress would slowly unravel the delicate architecture you explored earlier. So the leaf prepares. Protection does not begin when damage occurs. It begins in anticipation. Within the thylakoid membranes, special pigments stand ready. Among them are carotenoids, orange and yellow molecules you sometimes glimpse in autumn leaves when chlorophyll fades. During intense light, these pigments play a critical protective role. When excess energy builds within chlorophyll molecules, carotenoids accept some of that energy and release it safely as heat. This process is known as non-photochemical quenching. The name sounds technical, but the action is simple. Instead of allowing energy to cascade into harmful reactions, the plant converts it into warmth, a harmless vibration dissipating into the surrounding air. It is like loosening your shoulders when tension rises. You imagine the chloroplast breathing out tiny sighs of heat, invisible but essential. This protective mechanism activates quickly when light intensity rises. Proteins within the thylakoid membrane shift shape slightly. The arrangement of pigments changes. The system becomes less efficient at transferring energy to reaction centers, deliberately so. Efficiency decreases to preserve integrity. Here, you notice a paradox. To survive intense light, the plant must intentionally reduce how much light it uses for photosynthesis. It chooses safety over maximum output. This is called photoprotection. If protection were absent, the photosystem too complex, one of the primary light-absorbing structures, would suffer damage. In fact, even with protection, some degree of damage occurs daily under strong sunlight. The D1 protein within photosystem 2 is particularly vulnerable. It must be regularly removed and replaced in a constant cycle of repair. Photosynthesis is not a static engine. It is a living system requiring maintenance. You begin to understand that high light does not simply increase productivity in a straight line. Instead, productivity rises with light intensity only up to a certain point. Beyond that, gains slow. Eventually, too much light can reduce overall photosynthetic efficiency. This phenomenon is called photoinhibition. Photoinhibition does not mean the plant stops entirely. It means that damage outpaces repair. The system falters under pressure. You think of standing in bright sunlight too long without shade. Even warmth can become overwhelming. Leaves in natural environments rarely experience perfect conditions. Sunflecks break through forest canopies in shifting patterns. Clouds move unpredictably. Wind changes leaf angles moment by moment. Plants evolve to handle fluctuation, not constancy. Under extreme brightness, chloroplasts even reposition themselves within cells. Instead of lining the cell walls facing the sun, they move to the sides, reducing direct exposure. This movement is subtle, driven by changes in light intensity detected by photoreceptors. The cell rearranges its internal lanterns to avoid glare. And still, light continues to pour down. Water availability now becomes intertwined with light intensity. On hot, bright days, stomata, those tiny pores on the leaf's surface, may close partially to conserve water. But closed stomata limit carbon dioxide entry. With less carbon dioxide available, the Calvin cycle slows. Yet the light reactions may still be absorbing photons. This imbalance increases the risk of reactive oxygen formation. Protection must intensify. You begin to see how systems overlap. Excess light alone is challenging. Excess light combined with drought or heat becomes more dangerous. The leaf stands at the intersection of many forces. Even temperature plays a role here. Enzymes that repair damaged proteins work more efficiently within certain temperature ranges. Too cold, and repair slows. Too hot, and proteins themselves destabilize. Protection is costly. Energy used for heat dissipation is energy not stored as sugar. Resources devoted to repair cannot simultaneously fuel growth. The plant must allocate carefully. You might think that capturing every available photon would maximize survival. But evolution suggests otherwise. Plants that attempted to harvest light without restraint would suffer catastrophic damage during sudden bursts of brightness. Instead, natural selection favored systems that throttle output when necessary. Restraint becomes strategy. As afternoon drifts toward evening, the intensity of sunlight softens. Photoprotective mechanisms gradually relax. Reaction centers operate more efficiently again. Heat dissipation decreases. The leaf recovers. You realize that photosynthesis is not about constant maximization. It is about dynamic balance. It is about adjusting continuously to shifting conditions. Too little light starves the system. Too much light strains it. In between lies a moving target, a zone where energy flows steadily without overwhelming the machinery. And yet, even when light intensity is perfect, even when protection is balanced and damage minimal, another quiet limitation waits. Because light alone cannot build sugar. Energy captured in ATP and NADPH must be paired with carbon drawn from the air in invisible molecules drifting through tiny pores. As the sun lowers slightly and brightness becomes gentle once more, you feel your attention shifting from the sky above to the atmosphere itself. From photons to breath. From light's abundance to carbon's slower, quieter dance. The light is still present, but now your attention softens. You turn away from the brightness above and towards something almost invisible, the air itself. Around you, carbon dioxide drifts quietly. It makes up only a small fraction of the atmosphere, about 4 molecules in every 10,000. You cannot see it. You cannot feel it on your skin. And yet without it, all the captured light inside the chloroplast would remain unfinished potential. Energy alone cannot build sugar. For that, carbon is required. You move to the underside of the leaf, where tiny pores open and close like slow breaths. These are the stomata. Each stomata is bordered by 2 curved guard cells that swell or shrink depending on water pressure inside them. When they swell, the pore opens. When they relax, it closes. Through these openings, carbon dioxide enters. It diffuses inward, following a gradient from higher concentration in the air to lower concentration inside the leaf. There is no pump drawing it in. No active pulling force. Just quiet diffusion. Already, you sense limitation. Because diffusion is slow. Carbon dioxide must weave its way through air spaces between cells before finally dissolving into the watery interior of a chloroplast. Only then can it participate in the next stage of photosynthesis, carbon fixation. Inside the stroma of the chloroplast, a cycle unfolds. It is often called the Calvin cycle, named after the scientist who mapped its steps. But here, it feels less like a cycle and more like a patient workshop. The energy carriers created during the light reactions ATP and NADPH now offer their stored power. But they cannot build sugar alone. They need carbon atoms to assemble. And so enters rubisco. Ribulose 1,5-bisphosphate carboxylase/oxygenase. The name is long. The work is simple, yet profound. Rubisco is the enzyme that attaches carbon dioxide to a 5-carbon molecule called RuBP. This reaction begins the process of converting inorganic carbon into organic molecules that will eventually become glucose. Rubisco is the most abundant enzyme on Earth. And yet, it is surprisingly slow. Each rubisco molecule can fix only a few carbon dioxide molecules per second, perhaps 3 or 4 under ideal conditions. Compared to many enzymes that process thousands of reactions per second, rubisco works at a gentle pace. You imagine a careful craftsperson, assembling pieces deliberately rather than rushing. Why so slow? Partly because carbon dioxide is a stable molecule. Its bonds are strong. Attaching it to another molecule requires precision. Rubisco must position both CO2 and RuBP exactly right for the reaction to occur. And here, another complication appears. Rubisco cannot perfectly distinguish between carbon dioxide and oxygen. Both molecules are similar in size and shape. Both are present inside the chloroplast. Sometimes, instead of attaching CO2 to RuBP, rubisco binds oxygen instead. This alternative reaction initiates a process called photorespiration. Photorespiration consumes energy and releases previously fixed carbon. It does not produce sugar. It is, in a sense, an error. You might wonder why evolution would tolerate such inefficiency. The answer lies in history. Rubisco evolved billions of years ago when Earth's atmosphere contained very little oxygen. In that ancient world, confusion between CO2 and O2 was rare. But as photosynthetic organisms filled the atmosphere with oxygen, rubisco's limitation became more pronounced. The enzyme did not fundamentally change. And so, even today, a significant fraction of rubisco's activity leads to oxygen fixation rather than carbon fixation, especially when carbon dioxide levels inside the leaf are low. This means that even with abundant sunlight and fully functioning chloroplasts, sugar production can be limited by the availability of carbon dioxide and the selectivity of a single enzyme. You feel the rhythm slowing further. Carbon fixation is not explosive. It is iterative. Each turn of the Calvin cycle fixes one carbon atom. To build one molecule of glucose, which contains 6 carbons, the cycle must turn multiple times. ATP is spent. NADPH donates electrons. Intermediates rearrange. Enzymes guide each step. And all of it depends on the steady arrival of CO2 through stomata. But stomata cannot remain open without consequence. When they open, water vapor escapes from the leaf. This process is called transpiration. On warm or dry days, water loss can be substantial. If water becomes scarce, guard cells lose pressure and stomata close to conserve moisture. When stomata close, carbon dioxide entry slows dramatically. Light reactions may still be active. ATP and NADPH may accumulate. But without CO2, the Calvin cycle cannot proceed efficiently. The system becomes unbalanced again. You begin to understand that carbon dioxide concentration inside the leaf directly affects photosynthetic rate. If external CO2 levels rise, as they have gradually due to human activities, photosynthesis in some plants can increase, at least temporarily. But even here, there are limits. Rubisco becomes saturated at certain CO2 concentrations. Beyond that, additional carbon dioxide does not significantly increase fixation rate. Other factors, such as enzyme availability, temperature, or nutrient supply, become limiting. Photosynthesis is governed by the slowest essential step. Light capture may be rapid. Electron transport may be efficient. But if carbon fixation proceeds slowly, overall productivity slows with it. You sense how carefully balanced the entire system must be. Carbon dioxide enters quietly. Rubisco binds patiently. Sugars form gradually. Nothing is rushed. And even inefficiency has a kind of logic. Photorespiration, though costly, may help dissipate excess energy under certain conditions, protecting the chloroplast from damage when CO2 levels are low and light is high. What appears wasteful may serve subtle roles in stability. As afternoon light softens and the air cools slightly, stomata adjust. Guard cells respond to signals: light intensity, internal carbon concentration, water status, even hormonal cues like abscisic acid during drought. The leaf is constantly negotiating. You feel the quiet truth settling in. Capturing light is only half the story. The transformation of that light into sugar depends on invisible gases, delicate enzymes, and the slow choreography of diffusion. Abundance above does not guarantee abundance within. Carbon's dance is measured, intentional, bound by chemistry. And even as CO2 drifts inward through open pores, another partner remains essential. Because every time water molecules are split in the light reactions, every time stomata open to invite carbon in, water itself is moving, rising from soil through roots, climbing against gravity in narrow tubes. Without that steady ascent, the dance would falter. And so your attention begins to travel downward now, beneath the leaf, beneath the stem, toward the quiet pull of water rising from the earth. You let your awareness drift downward now. Away from the leaf. Down the stem. Into the roots that anchor the plant in darkness. Here, in the soil, water waits. It clings to tiny particles of clay and sand. It fills microscopic spaces between grains of earth. After rain, it gathers generously. During drought, it retreats into thin films, harder to reach. Roots do not drink in the way you might imagine. They absorb. Along the surface of young roots are countless fine root hairs, each one increasing the area available for contact with soil water. Through these delicate extensions, water enters by osmosis, moving from regions of higher water potential in the soil into lower water potential inside root cells. There is no pump pushing it upward from below. Instead, water is drawn upward by tension created in the leaves above. You return gently to the leaf, where sunlight still falls. As stomata open to allow carbon dioxide to enter, water vapor escapes into the air. This evaporation from the moist interior of the leaf creates a pulling force. Imagine a long chain of water molecules, each one lightly bonded to the next through hydrogen bonds. As molecules evaporate from the leaf surface, they tug on the ones below. That pull extends down through the stem, through narrow tubes called xylem vessels, all the way to the roots. This is called the cohesion tension mechanism. It is elegant and silent. Water climbs upward against gravity not because it is pushed, but because it is pulled. You picture columns of water rising through the plant like transparent threads, unbroken from root to leaf. The narrowness of xylem vessels helps maintain these columns. The adhesion of water molecules to the vessel walls stabilizes the ascent. And this ascent is essential. Inside the chloroplast, during the light reactions, water molecules are split. This process, called photolysis, occurs within photosystem too. Light energy extracts electrons from water, producing oxygen, protons, and electrons. Those electrons replace the ones lost by chlorophyll when photons excite them. Without water, the chain would stall at the very beginning. Water is not only a solvent. It is a donor of electrons. Each time oxygen drifts from a leaf into the atmosphere, it carries the memory of water split by light. But water's role extends further. When stomata open for carbon dioxide to enter, water inevitably escapes. This trade-off is unavoidable. The same pores that allow carbon and allow water out. On mild days, when soil moisture is plentiful, this exchange proceeds smoothly. Water lost through transpiration is replenished continuously from below. The pull remains steady. But when soil dries, the balance shifts. Water potential in the soil drops. Roots struggle to extract what remains. The tension in xylem vessels increases. If tension becomes too great, air bubbles can form in the water column, a process called cavitation. These bubbles block water transport. The ascent falters. Sensing water stress, the plant responds. Guard cells lose turgor pressure. Stomata begin to close. Carbon dioxide entry slows. Photosynthesis dims. You feel the interdependence clearly now. Light may be abundant. Chloroplasts may be intact. Rubisco may be ready. But without sufficient water, the entire system contracts. Water is both partner and gatekeeper. It enables electron flow. It sustains turgor pressure in cells, keeping leaves upright and oriented toward light. It allows stomata to open safely. It cools the leaf through evaporative loss, preventing overheating under intense sunlight. Transpiration can lower leaf temperature by several degrees. Without it, heat would accumulate, increasing the risk of protein denaturation and photodamage. And yet, every droplet lost must be replaced. In arid environments, plants evolve strategies to conserve water. Some develop thick cuticles, waxy coatings that reduce evaporation. Others reduce leaf size or transform leaves into spines, minimizing surface area. Some plants open their stomata at night instead of day, a strategy known as CAM photosynthesis. By fixing carbon dioxide in the cooler, more humid nighttime air, they reduce water loss. Each adaptation reflects the same truth. Water availability constrains photosynthesis. Even in temperate forests, temporary drought can reduce carbon assimilation significantly. Stomatal closure protects against dehydration but restricts carbon intake. Internal carbon dioxide concentration drops. Rubisco increasingly fixes oxygen instead of carbon, increasing photorespiration. Light reactions may continue, but the Calvin cycle slows. The balance you have been tracing between light, carbon, and structure now reveals another axis. Hydration. You imagine the plant at midday during a dry spell. The sun blazes overhead. Soil moisture dwindles. Stomata narrow. Chloroplasts receive light but cannot fully use it. Protective mechanisms activate to dissipate excess energy as heat. Again, safety reduces productivity. Water scarcity does not merely pause growth. It reshapes the entire flow of energy within the leaf. And yet, when rain returns, something beautiful happens. Soil absorbs moisture. Roots rehydrate. Tension in xylem eases. Guard cells swell once more. Stomata reopen. Carbon dioxide diffuses inward. The quiet ascent resumes. Photosynthesis brightens again. You realize that the plant is always negotiating between two risks, desiccation and starvation. Open too wide, and water is lost too quickly. Close too tightly, and carbon cannot enter. Balance is never fixed. As evening approaches and temperatures cool, transpiration slows naturally. The pull on water columns softens. Stomata may close gradually as light fades. Darkness brings temporary rest to the light reactions, but water continues its subtle movements within tissues, redistributing, stabilizing. You feel how deeply integrated this system is. The sun shines above. Carbon drifts in the air. Water rises from the earth. And the leaf stands at their meeting point, managing exchange with quiet precision. But even when water is plentiful, even when light is moderate and carbon sufficient, another invisible variable shapes the pace of life. Because the speed of molecules, the flexibility of proteins, the rhythm of reactions all depend on temperature. And as the warmth of afternoon lingers around the leaf, you begin to sense how heat itself sets the tempo for everything that follows. You remain with the leaf, but now you notice something subtler than light, quieter than water. You notice warmth. The afternoon air wraps around the plant in a gentle embrace. The sun has traveled high and begun its slow descent. The brightness is softer than noon, but the heat lingers. Temperature is not something you can see. It is motion. It is the speed at which molecules tremble and drift. When warmth increases, atoms vibrate more quickly. When cold settles in, movement slows. Inside the chloroplast, inside the stroma where carbon fixation unfolds, temperature shapes everything. Enzymes, those intricate protein molecules guiding each reaction, depend on motion. They must bend slightly, flex gently, position substrates precisely. Their shape is not rigid like stone. It is dynamic, responsive. You imagine an enzyme as a pair of careful hands. When the temperature is just right, the hands move with confidence. They grasp carbon dioxide and RuBP, align them perfectly, and guide them into reaction. The product forms, and the hands release it calmly. But when the air turns cold, something changes. Molecules move more slowly. Collisions between enzyme and substrate become less frequent. Even when they meet, the reduced kinetic energy makes it harder to overcome the activation barrier required for reaction. The hands feel stiff. Rubisco, already slow by nature, becomes slower still. The Calvin cycle turns gently, almost reluctantly. ATP and NADPH may accumulate because the light reactions are less sensitive to moderate cold than the carbon-fixing enzymes are. Photosynthesis declines. You sense the quiet of a cool morning in early spring. The sun shines brightly, yet the air remains crisp. The leaf absorbs light, but the internal chemistry cannot keep pace. Energy waits. Now imagine the opposite. The air grows hot. Not just warm, but heavy. Molecules vibrate rapidly. Collisions become more frequent, more forceful. At first, reaction rates increase. Within a moderate temperature range, photosynthesis often rises with warmth. Enzymes work more quickly. Diffusion accelerates. Metabolic pathways hum with energy. But only up to a point. Proteins are held together by delicate bonds, hydrogen bonds, ionic interactions, hydrophobic forces. Excessive heat disrupts these interactions. The precise three-dimensional structure of an enzyme begins to loosen. This process is called denaturation. The careful hands lose their shape. When an enzyme denatures, its active site, the specific region where substrates bind, changes form. Carbon dioxide may no longer fit properly. RuBP may not align correctly. Reaction slows, then falters. Rubisco is sensitive to temperature in complex ways. At higher temperatures, its ability to distinguish between carbon dioxide and oxygen decreases. Photorespiration increases, diverting energy away from sugar production. You see how heat not only threatens structure but alters balance. Membranes, too, respond to temperature. Thylakoid membranes become more fluid as warmth increases. If they become too fluid, the precise organization of photosystems can destabilize. Electron transport chains may become less efficient. Leakage of protons across membranes can reduce the gradient needed to produce ATP. Structure and temperature are inseparable. On extremely hot days, leaves may overheat beyond air temperature because they absorb sunlight. Transpiration helps cool them, but if water is scarce and stomata close, cooling diminishes. Internal temperatures can rise to damaging levels. Plants have evolved heat shock proteins, special molecules that help stabilize and refold damaged proteins under stress. These are emergency responders within the cell. But emergency responses consume energy. You begin to feel how narrow the optimal range truly is. Most temperate plants perform best within a span perhaps between 20 and 30 degrees Celsius. Outside this range, efficiency drops gradually, then sharply. Tropical plants may tolerate higher temperatures, alpine plants lower ones. Each species carries its own thermal preferences, shaped by evolution in particular climates. And yet, even within that range, daily fluctuations matter. At night, as temperatures fall, metabolic rates slow. During the day, warmth accelerates them again. This diurnal rhythm influences how much carbon is fixed, how much sugar is stored, how much growth occurs. Temperature does not act alone. It interacts with light and water. High light combined with high temperature increases the risk of photoinhibition. High temperature combined with drought amplifies stress, as stomata close and cooling decreases. Conversely, cool temperatures may reduce water loss but also slow carbon fixation. You begin to sense a web of dependencies. Photosynthesis is not governed by a single dial. It is shaped by many dials turning together: light intensity, carbon availability, hydration, and now heat. Enzymes move within this thermal landscape like dancers adjusting to music. Too slow, and the dance drags. Too fast, and steps become chaotic. Some plants acclimate over time. They adjust enzyme concentrations. They modify membrane lipid composition to maintain appropriate fluidity under different temperatures. Acclimation allows partial compensation. But no plant escapes the fundamental truth. Life operates within a limited thermal window. You imagine the leaf resting in the warmth of late afternoon. Reactions proceed steadily. Rubisco binds carbon dioxide with patient rhythm. ATP forms and is consumed. Water rises quietly. The system hums. Then clouds drift in, shading the leaf. Temperature drops slightly. The rhythm slows. Later, a heat wave arrives. The air thickens. The plant activates protective responses once more. Temperature sets the tempo. You feel how finely tuned this orchestration must be. Each enzyme shaped by evolution to function within a particular range. Each membrane composed of lipids selected for stability at certain temperatures. And beyond these internal constraints lies another layer, something drawn from the soil, something incorporated into chlorophyll itself. Because even if light is perfect, water sufficient, carbon abundant, and temperature ideal, the plant still depends on elements it cannot create. Nitrogen, magnesium, iron. Without them, enzymes cannot form properly. Chlorophyll cannot assemble. Electron transport falters. You sense your attention beginning to sink once more downward, beneath the roots, into the quiet darkness where minerals wait unseen. You travel downward again. Past the stem. Past the branching roots. Into the soil where light does not reach. Here, beneath the surface, life continues in shadow. Particles of sand, clay, and organic matter press together in intricate patterns. Between them lie thin films of water carrying dissolved minerals, the quiet ingredients of growth. Sunlight may power photosynthesis, but it cannot build the machinery alone. For that, the plant depends on elements drawn from the earth. Nitrogen is one of the most important. It is a core component of amino acids, the building blocks of proteins. Enzymes like Rubisco are constructed from long chains of amino acids folded into precise shapes. Without nitrogen, those chains cannot form. And Rubisco, you remember, is the most abundant enzyme on Earth. Nitrogen is also part of chlorophyll itself. At the center of every chlorophyll molecule lies a ring structure containing nitrogen atoms arranged carefully around a magnesium ion. This ring allows chlorophyll to absorb light efficiently. Without nitrogen, the leaf pales. Chlorophyll production drops. Photosynthesis weakens. You imagine a plant growing in nitrogen-poor soil. The light shines brightly above, but the leaves appear yellowish, a condition called chlorosis. The machinery is incomplete. The system lacks pieces. Nitrogen enters the plant primarily as nitrate or ammonium ions dissolved in soil water. Root cells actively transport these ions inward. Energy is required. Uptake is not passive. Once inside, nitrate must be reduced to ammonium before it can be incorporated into amino acids. This reduction consumes energy that originally came from light. Even gathering nutrients has a cost. Magnesium is another essential element. It sits at the heart of the chlorophyll molecule, stabilizing its structure. Remove magnesium, and chlorophyll cannot function properly. Light absorption falters. Magnesium also plays roles in stabilizing ribosomes, the cellular machines that build proteins, and in activating certain enzymes. Iron, though needed in smaller quantities, is equally vital. It participates in electron transport within both photosystems and in the formation of cytochromes, proteins that shuttle electrons during the light reactions. Without iron, the electron transport chain weakens. Energy transfer slows. You begin to sense how dependent the chloroplast is on these invisible ions drawn from the soil. And soil is never uniform. Its nutrient content varies with geology, organic matter, microbial activity, rainfall, and human intervention. Some soils are rich and dark, full of decomposed plant material releasing nitrogen and phosphorus. Others are sandy and sparse, holding little beyond mineral grains. Plants growing in nutrient-poor soils often adapt in subtle ways. They may produce fewer leaves. They may grow more slowly. Some form symbiotic relationships with fungi and mycorrhizae that extend the effective reach of their roots, helping them access nutrients more efficiently. These fungal networks weave through soil like hidden threads, exchanging minerals for sugars produced by the plant. Cooperation emerges from limitation. You reflect on phosphorus, another critical nutrient. Phosphorus is part of ATP, the energy currency of the cell. Without phosphorus, ATP cannot form. Energy captured from light would lack a vehicle for storage and transport. Phosphorus is also part of DNA and RNA. It anchors the backbone of genetic information. And yet phosphorus in soil often binds tightly to particles, becoming difficult for roots to access. Plants must release organic acids or enzymes to free it. Again, effort is required. Even potassium, though not incorporated into organic molecules in the same way, is essential for regulating stomatal opening. Guard cells rely on potassium fluxes to adjust their turgor pressure. Without adequate potassium, stomatal function becomes impaired. Carbon dioxide entry suffers. Photosynthesis declines. You feel how interconnected everything is. Light may be abundant. Water may rise steadily. Temperature may fall within optimal range. But if nitrogen is scarce, rubisco concentration drops. If magnesium is lacking, chlorophyll weakens. If iron is insufficient, electron flow stumbles. Photosynthesis slows not because the sun fails, but because the soil cannot supply. Nutrient limitation often reveals itself gradually. Leaves may grow smaller. Growth rates diminish. The plant reallocates resources, perhaps prioritizing root growth to explore more soil in search of what is missing. In agricultural systems, humans add fertilizers to replenish depleted soils. This can dramatically increase photosynthetic capacity, but only up to a point. Beyond certain concentrations, additional nutrients do not proportionally increase productivity. Other factors become limiting. This principle is often described by the law of the minimum, first articulated by Justus von Liebig. It suggests that growth is controlled not by the total resources available, but by the scarcest essential resource. You imagine a wooden barrel made of uneven staves. The shortest stave determines how much water the barrel can hold. So it is with plants. Even if 9 conditions are favorable, the 10th, if deficient, constrains the whole. In natural ecosystems, nutrient cycling is continuous. Leaves fall and decompose. Microorganisms break down organic matter, releasing nitrogen and phosphorus back into the soil. Roots absorb them again. The dance between leaf and soil repeats. You picture the forest floor in autumn, covered with fallen leaves. What once captured light now returns to darkness, slowly decomposing. Nutrients are freed. The cycle continues. Nothing is wasted, but nothing is limitless. You begin to feel the quiet humility of the leaf. It depends on what lies beneath it. Its green glow is not only a reflection of sunlight but of soil chemistry. And even when nutrients are sufficient, light is still unevenly distributed across the canopy. Some leaves bask in full sun. Others linger in shadow cast by neighbors. Shape and arrangement begin to matter. As you rise gently upward from the soil, back through roots and stems, you look toward the canopy once more. Because beyond chemistry and temperature and water, there is geometry, the way leaves position themselves to meet the sun, and the way light itself is shared. You lift your awareness once more, leaving the soil behind. Now you rise above the forest floor, above the stems and branches, and into the canopy itself. From this perspective, the leaves transform into a mosaic, a quilt of green with patterns that seem deliberate yet effortlessly natural. Each leaf tilts, turns, and spreads in directions that maximize the capture of light, yet never entirely dominates the space. You notice that no two leaves are identical in position or angle. Sunlight touches some directly, striking at near-perfect perpendicularity. Others receive dappled patches through the gaps above. Shadows shift as the wind rocks branches, and as the sun arcs slowly across the sky, these patterns change minute by minute. Light is abundant, yet never uniform. The geometry of leaves is a conversation with this shifting abundance. Broad leaves may stretch flat to catch sunlight efficiently in shady understories. Needle-like leaves, like those of pines, reduce surface area to avoid excessive exposure in intense light. Compound leaves spread their leaflets to reduce self-shading while still intercepting photons. Each leaf becomes both participant and negotiator, balancing opportunity and restraint. You watch as plants interact through the language of angles. Taller trees overarch shorter neighbors, creating shade below. Some plants grow vertically, narrow, or climb on supports to reach brighter patches. Others spread horizontally, maximizing surface area where light is less intense. Competition is quiet but constant. Yet cooperation exists too. When leaves overlap, they create a canopy that reduces extremes of temperature and water loss. Shadows can shelter understory plants from desiccation, enabling life to thrive beneath. The geometry of leaves is not only a strategy for photosynthesis but also a subtle management of environmental stress. You imagine standing atop the canopy, feeling sunlight and shade play across your skin in changing patterns. The leaves you see are not passive, they are sensors, receivers, and transmitters, adjusting continually to the movements of the sun. They twist, rotate, and even reposition chloroplasts within their cells to optimize photon capture while preventing damage from overexposure. This positioning is called heliotropism, in some species a slow rotation of leaves to track the sun. Others maintain fixed orientations but adjust the angle of individual leaflets, responding to local light intensity and internal signaling. Shape and angle determine how much light each leaf can use. It is a negotiation written in geometry. Beyond individual leaves, patterns emerge at the scale of branches and the whole tree. Leaf arrangement along a stem follows mathematical principles that reduce self-shading. In many species, leaves spiral along the stem in a pattern called phyllotaxy. This spiral distributes leaves so that each has its own access to light while minimizing overlap. You feel the elegance of this arrangement. Mathematics and biology entwined, allowing life to capture light efficiently without wasting energy. Yet even with perfect geometry, light is not guaranteed. Clouds drift overhead. Neighboring trees sway in the wind. Seasonal changes alter the sun's path. Leaves must be flexible and adaptive. Geometry provides structure, but not certainty. The shape of each leaf also reflects trade-offs. Broad leaves may intercept more light but are more vulnerable to overheating and water loss. Small, Thin leaves reduce these risks but capture less energy per unit area. The leaf's geometry must balance light capture, water conservation, temperature regulation, and even resistance to herbivory. You notice the interplay of form and function. Even within a single tree, leaves differ. Lower leaves in shade may be broader and thinner. Upper leaves in full sun may be smaller, thicker, and more rigid. The plant fine-tunes its geometry in response to the environment, a subtle dialogue between structure and necessity. You begin to appreciate light as a currency abundant yet rationed by shape, position, and environmental context. No leaf can capture all that falls upon it. The geometry of the canopy ensures that light is shared unevenly, creating a dynamic mosaic of abundance and scarcity. And yet, even with perfect positioning and clever angles, not all potential is realized. Limits imposed by carbon dioxide availability, water supply, temperature, and nutrients persist. Leaves can capture light efficiently, but they cannot overcome shortages in other resources. The geometry of leaves, elegant as it is, exists within the broader network of constraints. The architecture of the canopy interacts with the internal architecture of cells, the rhythm of enzymes, and the availability of essential molecules. You float slowly over the canopy, observing leaves tilting and turning as if aware of the subtle dance between light and limitation. The forest breathes in response to sunlight, water, and carbon. Leaves sway, adjusting angles, capturing photons, dissipating excess energy, and sending sugars down to roots. Patterns repeat across scales. From chloroplasts to cells, cells to leaves, leaves to branches, branches to trees, the interplay of geometry shapes the efficiency of photosynthesis. It is a multi-layered puzzle, where structure, orientation, and timing all converge. You feel a sense of rhythm, a balance maintained through shape, movement, and constraint. Geometry is not merely physical, it is functional, adaptive, and responsive. As the sun continues its journey westward, shadows lengthen. The angle of light shifts. What was once fully illuminated now rests in gentle shadow. Leaves adjust, some curling slightly to reduce exposure, others orienting to catch the fading sun. And you sense that geometry is just one piece of the story. Within the cells, reactions continue, carbon is fixed, electrons flow, and water moves silently. Above, light is partitioned by form and position. Beneath, nutrients and water rise to sustain the structure. Yet another subtle limitation is always present. Even perfect leaf arrangement cannot prevent the inefficiencies of the system. Rubisco still misfires. Enzymes respond to temperature. Water availability fluctuates. Minerals are finite. You rise higher, above the canopy, sensing the intricate choreography between sunlight and green surfaces, between geometry and chemistry, between opportunity and restraint. And now, as your gaze softens, your attention drifts inward once more, toward the enzymes within the chloroplast, returning to the microcosm where tiny mistakes ripple outward, where rubisco sometimes binds oxygen instead of carbon, and the dance of light and life continues with all its beautiful inefficiency. You descend now into the chloroplast, into the intimate chambers where light energy has already been captured and carbon dioxide has arrived through the stomata. Rubisco, the patient enzyme, waits quietly within the stroma. It is the most abundant enzyme on Earth, yet it is prone to a subtle confusion, a gentle, recurring imperfection. Carbon dioxide drifts into the active site, ready to join with RuBP, beginning the process of sugar formation. But sometimes, oxygen slips in instead. Rubisco does not discriminate perfectly. Its structure evolved in an ancient world when oxygen was scarce, and so the enzyme's memory carries that history. Today, in an oxygen-rich atmosphere, this flaw manifests daily. This mistake initiates a process called photorespiration. It is called a wasteful pathway because energy and previously fixed carbon are lost. Yet the term wasteful feels inadequate when you look closer. There is subtlety here, a deeper rhythm. When oxygen binds, the resulting molecule cannot continue through the Calvin cycle. Instead, it enters a series of reactions spanning multiple cellular compartments, the chloroplast, the peroxisome, and the mitochondrion. Molecules are transformed, rearranged, and recycled. Carbon that was almost fixed is released as carbon dioxide, and ATP is consumed in the process. At first, this seems like an inefficiency, a system countering its own efforts. Energy used for photosynthesis has been partially undone. Sugar yield drops. But even inefficiency has a purpose. Photorespiration helps dissipate excess energy within the chloroplast when carbon dioxide is limited or when light intensity is high. Without it, reactive oxygen species could accumulate, damaging photosystems and membranes. In essence, this mistake acts as a safety valve, protecting the leaf from harm. You imagine Rubisco at work, making occasional errors, and yet each error ripples gently through the system. The plant does not collapse. Instead, it maintains balance. Protection and energy management coexist with production. Imperfection becomes a tool for survival. The steps of photorespiration are intricate. Glycolate, produced when oxygen binds to RuBP, exits the chloroplast. It travels to the peroxisome, where it is converted to glyoxylate. Hydrogen peroxide is formed as a byproduct, which is carefully broken down by catalase enzymes. Then the molecules move to mitochondria, where energy is expended to convert them back into forms usable in the Calvin cycle. The journey is long and deliberate. Carbon is partially lost, energy is spent, yet nothing catastrophic occurs. Life adapts. Life persists. You notice a rhythm here. The enzyme's error, the recycling of carbon, the delicate balancing act of energy, it is almost meditative. A constant correction, a self-regulating cycle, like a quiet tide smoothing stones on a shore. Imperfection is not chaos, it is rhythm, history, and compromise. Some plants have evolved strategies to reduce photorespiration. C4 plants, like maize and sugarcane, concentrate carbon dioxide around Rubisco, minimizing oxygen's interference. Cam plants fix carbon dioxide at night to reduce water loss and increase CO2 availability during the day. Yet even these adaptations cannot eliminate the phenomenon entirely. The persistence of photorespiration reminds you that evolution favors balance over perfection. Maximum output is not always ideal if it carries risk. Rubisco's mistake is an ancient compromise, retained because it contributes to resilience. You visualize the leaf once more. Sunlight streams down. Carbon dioxide diffuses in. Water climbs from the roots. Rubisco works with gentle imperfection, catalyzing reactions that sometimes require correction. Photorespiration consumes energy, but it also preserves the machinery that allows photosynthesis to continue. This inefficiency is beautiful in its subtlety. Without it, the chloroplast could be damaged under intense light. Without it, the entire process might be more brittle, less capable of adjusting to fluctuations in light, temperature, and water. Nature tolerates a small loss to maintain the whole system. You sense a pattern emerging across scales. Earlier, you observed geometry dictating light capture. Then water controlled the rhythm of stomata, temperature set the pace for enzymes, and nutrients determined how much chlorophyll could form. Now, within the chloroplast, Rubisco's error adds another layer, a microscopic limitation that protects the larger system. Life is constrained, yet resilient. You think of the forest canopy. Each leaf is imperfect, yet collectively they sustain the ecosystem. Individual errors are buffered by redundancy and integration. Imperfection is woven into the architecture of survival. As the day progresses, light intensity fluctuates. Photorespiration rises during the brightest hours, when carbon dioxide becomes limiting relative to oxygen. Then, as the sun lowers, temperature cools slightly, and stomata adjust, the rate declines. The plant moves through these shifts smoothly, without sudden disruption. You notice that energy and carbon flow in a delicate dance. ATP and NADPH are produced by light reactions, then consumed in carbon fixation. Rubisco's occasional mistake slows the flow, but the system adapts. Protective mechanisms and repair processes compensate. Productivity continues, not at maximum speed, but steadily, sustainably. You reflect on imperfection. In human terms, mistakes are often seen as failures. But in the leaf, a controlled mistake allows survival. It prevents catastrophic damage. It balances multiple priorities: efficiency, safety, and adaptability. Photorespiration is a reminder that nature values stability over perfection. It is an ancient strategy, shaped over billions of years, preserved because it works. The system tolerates minor losses to maintain continuity. And yet, you wonder, if Rubisco had evolved perfect specificity, would life have been faster, more productive? Perhaps. But perhaps the system would be more fragile, less able to adjust to sudden fluctuations in light, water, or temperature. The flaw may be essential. You float within the chloroplast, observing molecules moving, reactions unfolding, electrons passing along chains. Energy is both created and carefully managed. Carbon is fixed, sometimes lost, and recycled. The leaf operates not in perfection, but in balance. Even inefficiency has a place in the symphony of life. Each misstep creates space for stability, protection, and resilience. Every photorespiratory cycle is a whisper of ancient conditions, a reminder of the atmosphere's history, and a lesson in compromise. And so you watch, feeling the gentle cadence of this beautiful inefficiency, noticing how life tolerates small errors to maintain the continuity of something much larger: the leaf, the plant, the forest, and the invisible threads connecting all energy, carbon, and water. As sunlight softens toward evening, and leaves adjust their angles for the waning rays, you sense that even as the day concludes, the subtle dance of imperfection continues. Rubisco, photorespiration, energy flow all remain poised, ready for tomorrow's light. And in that quiet anticipation, you feel drawn toward the broader picture: every limitation, every balance, every gentle constraint that together shapes why plants never capture all the light, yet thrive beautifully within the bounds nature has set. You rise once more above the forest canopy, this time seeing the scene not as individual leaves, not as enzymes in a chloroplast, but as the entire living system, the forest as a network of intertwined limitations and possibilities. Every leaf, every branch, every cell contributes to a symphony of constraints. Sunlight falls generously, but the canopy intercepts it unevenly. Some leaves bask in brilliance while others linger in shadow. You feel the dance of light and shade as a quiet rhythm rather than a precise measurement. Water rises silently from the soil, pulled by evaporation through leaves. But water is finite. Droughts or temporary shortages immediately ripple through the plant, slowing stomata, dimming photosynthesis, and altering the pace of life within each leaf. Carbon dioxide drifts in invisibly. It is necessary, yet limited. The slow, patient work of Rubisco reminds you that efficiency is bounded by chemistry itself. Even when light and water are abundant, carbon can be scarce inside the leaf, slowing production. Temperature shapes the tempo. Enzymes move with grace within a narrow thermal window, slowing in the cold, faltering in excessive heat. The delicate structures of proteins and membranes respond to warmth and chill alike, a rhythm finely tuned but not infinite in flexibility. Nutrients beneath the soil remain unseen but essential. Nitrogen, magnesium, iron, phosphorus, potassium, each pulled from the earth by roots, each determining whether the leaf can synthesize chlorophyll, build enzymes, and carry electrons through the photochemical pathways. Even sunlight cannot compensate for these unseen shortages. Photorespiration adds another layer, a reminder that even with all inputs present, energy is sometimes spent correcting what cannot be prevented. Inefficiency becomes protective, a buffer against extremes, a compromise between survival and productivity. You see these constraints overlapping like translucent veils. No single factor dominates entirely. No limitation acts in isolation. They intersect, interweave, and collectively define what is possible. Light alone cannot drive photosynthesis to infinite rates. Water, carbon, temperature, nutrients, and enzyme dynamics each set a boundary. Perfection, you realize, would demand infinite energy, infinite precision. Every enzyme perfectly selective, every leaf ideally positioned, every stomata perpetually open without water loss. Such a scenario is impossible. The physical world, chemical laws, and evolutionary history impose boundaries. Instead, life chooses balance. Plants capture enough light to grow, to reproduce, and to sustain ecosystems, but never all the light. Excess photons might overwhelm, water loss might threaten survival, temperature extremes might denature enzymes, nutrients might run short. The system responds by restraining itself, not to fail, but to endure. You reflect on this restraint as a form of wisdom. Nature does not pursue maximal output at any cost. Efficiency is tempered by safety, stability, and sustainability. Photosynthesis is not a race to the top. It is a measured negotiation among many factors, each pulling and constraining, each contributing to the whole. Leaves twist and turn, adjusting their angles. Stomata open cautiously. Water ascends through xylem only as the leaf requires. Rubisco binds CO2, occasionally misfiring, yet preserving the integrity of the chloroplast. Minerals flow slowly from the soil, incorporated into molecules only as needed. Every element acts within its own rhythm. Even in the brightest midday sun, the plant does not strive for maximal capture. Protective pigments dissipate excess energy as heat. Electrons flow along alternative pathways. Photorespiration removes surplus excitation. The leaf moderates its own potential. You sense that this moderation is not a failure but an elegance. Life operates within limitations not as a constraint but as a structure for possibility. Each boundary is a guideline for safe operation. Each inefficiency safeguards continuity. Each restraint allows flexibility and resilience. From the perspective of the canopy, the forest is a patchwork of efficiency tempered by moderation. Sunlight falls freely, yet not every photon is absorbed. Energy flows in measured steps through leaves, stems, branches, and roots. Carbon flows gradually, water moves patiently, sugars are distributed steadily. The system thrives not because it maximizes instantaneous output, but because it honors the limits imposed by physics, chemistry, and biology. You imagine a single leaf during midday. Sunlight pours down. CO2 is available. Water is sufficient. Temperature ideal. Yet the leaf absorbs only as much as it safely can. Enzymes pace themselves. Stomata balance carbon gain against water loss. Protective mechanisms intervene. The leaf operates not at the peak of possibility but at the peak of sustainable balance. Life, you realize, is composed of overlapping compromises. Photosynthesis is the clearest example you have seen. It is a process that depends on multiple constraints interacting in subtle ways, each moderating the other. Too much light without water causes stress. Ample carbon without proper temperature slows reactions. Perfect geometry without nutrients yields limited output. You reflect on how this principle extends beyond photosynthesis. In the forest, every organism, every interaction, is bounded by resources, energy, and risk. Survival depends not on achieving maximal potential at all times, but on negotiating limitations gracefully. Balance ensures continuity. Restraint allows resilience. The forest canopy sways gently in the afternoon breeze. Light filters through layers of leaves, creating patterns of brightness and shadow that shift continuously. Some leaves shine with full sunlight, others receive dappled patches. The rhythm is dynamic yet stable. Energy flows through the system in pulses and pauses, never fully exhausting, always adapting. You begin to understand why plants never capture all the light. Every absorbed photon is part of a larger negotiation, a subtle dialogue with water, carbon, temperature, nutrients, and enzyme kinetics. The leaf cannot act alone. It cannot overwhelm one boundary without respecting another. Perfect efficiency is impossible. Maximum capture is unattainable. And yet, this does not hinder life. Instead, it shapes a system that is durable, flexible, and harmonious. Each limitation becomes a stabilizing factor. Each boundary becomes a framework within which life can flourish. You watch sunlight slowly soften as the day advances. The forest transitions into evening. Shadows lengthen. Leaves adjust, reacting subtly to fading light. Stomata begin to close gradually. Water movement slows. Photosynthesis winds down without a sudden halt. The system prepares for the cooler, quieter hours of night. You feel the wisdom and the restraint. Nature chooses not maximum, but enough. Not constant acceleration, but measured rhythm. Not perfection, but balance. The overlapping limitations, light, carbon, water, temperature, nutrients, enzymatic constraints, are not failures. They are harmonies. And as the sun dips lower, casting a golden glow over the canopy, you sense the quiet truth of the forest. Life thrives not by capturing all, but by respecting the boundaries that make survival possible. The leaf, the branch, the tree, and the forest as a whole are composed of countless compromises, each essential, each protective, each beautiful. And as you let your attention drift downward once more, you begin to see the gentle mathematics that governs not only energy capture, but the balance of life itself, the calculation of enough, not too much, and never less than necessary. Tomorrow, you will rest with the sun at the horizon, reflecting on the delicate harmony between growth and sustainability, and the quiet lesson that limits are not failures, but the very foundation of enduring life. You drift gently with the evening air, the golden light softening into the violet hues of twilight. The forest is quieter now. The sun lowers, its rays grazing the treetops at an angle that illuminates some leaves and leaves others in gentle shadow. Photosynthesis continues, but more slowly. The light reactions pulse with diminishing intensity. Electrons move with less urgency. ATP and NADPH form more gradually, signaling a natural pause. Stomata adjust as the air cools, balancing water loss against carbon dioxide uptake. Enzyme activity slows with falling temperature, and the Calvin cycle moves more gently. You notice that the forest does not falter. Life persists. Leaves shimmer in the twilight, their chlorophyll still poised to capture whatever light remains. Roots continue to draw water and nutrients, albeit more slowly. The system is not racing to maximum efficiency, it is maintaining balance. This is the mathematics of enough. Through all the layers you have observed light, water, temperature, nutrients, geometry, enzyme efficiency, and the protective nuances of photorespiration, a pattern emerges. Life does not aim to capture every photon, to fix every molecule of carbon, or to grow at every possible moment. Instead, it negotiates, carefully and quietly, within the limits imposed by the world. Efficiency yields to stability. Growth bows to sustainability. Energy is invested thoughtfully, resources are allocated wisely, and imperfection is embraced as a safeguard rather than a failure. You reflect on the subtle interdependencies. A leaf does not exist in isolation. Its light capture depends on its neighbors' positions. Its water availability depends on soil moisture and root access. Its enzymatic efficiency depends on temperature, hydration, and nutrients. And its ultimate contribution to sugar production is moderated by the tiny errors of rubisco and the protective inefficiency of photorespiration. Every factor overlaps. Every constraint is interwoven. The plant, the tree, the forest all operate as a network, a system of compromises finely tuned by evolution over millions of years. You watch as shadows lengthen and the forest exhales in the cool evening breeze. Leaves sway, adjusting angles to catch the last light. Photosynthesis slows, but respiration continues, maintaining internal energy balances. Sugars produced during the day are transported to roots, stems, and developing seeds. Water movement decelerates, yet columns of xylem still hold tension, ready for the next morning. You sense the elegance of the system. Each constraint, once perceived as a limitation, now appears as a structure for harmony. Without the boundaries imposed by water, carbon, nutrients, temperature, and enzyme dynamics, life might push beyond its safe operating range, risking damage, inefficiency, or collapse. Nature prefers balance over extremity. You recall the geometry of leaves, the rise of water through xylem, the rhythm of enzymes, the slow dance of carbon fixation, the quiet imperfection of rubisco. Each chapter of the plant's story has contributed a layer to this final understanding. Light is abundant, yet leaves only capture what they can safely use. Water flows, yet drought imposes restraint. Nutrients provide building blocks, yet scarcity dictates priority. Temperature sets the pace, and inefficiencies within enzymatic reactions safeguard stability. All these threads converge in twilight. You close your attention on a single leaf. Its edges glow faintly in the lingering sunlight. You feel the subtle mathematics at work, the sum of limitations, the gentle calculations that prevent excess, the natural optimization that favors enough. Life thrives not by relentless accumulation, but by measured equilibrium. You reflect on the forest as a whole. Every tree, every branch, every leaf participates in this quiet arithmetic. Light is partitioned. Water flows according to demand. Carbon moves according to availability. Energy is allocated to protect as much as to build. The entire ecosystem is a mosaic of constraints, yet it is remarkably productive, resilient, and enduring. You sense a profound lesson: restraint is not a weakness. Boundaries are not failures. Limits are not shortcomings. They are harmonies, guiding life to operate sustainably within the possibilities of the environment. You think of the seasons, cycling endlessly. Spring brings growth, summer abundance, autumn harvest, and winter rest. Each phase respects limits: water availability, temperature, light intensity, and nutrient accessibility shift naturally. Plants do not resist these changes, they integrate them into their rhythms. Twilight deepens. The sun disappears behind the horizon, leaving a lingering glow that fades slowly into night. Photosynthesis ceases, yet the forest remains alive and poised for tomorrow. Sugars stored in roots and stems provide energy. Enzymes stabilize in preparation for cooler temperatures. Water redistributes within cells. Protective pigments dissipate residual energy. Even in the absence of sunlight, life is prepared to resume seamlessly at dawn. You feel a sense of completeness, yet it is not a static completion. The plant's story continues, moving with the cycles of light and dark, warmth and cool, abundance and limitation. Each day is a repetition of negotiations, compromises, and adjustments. The mathematics of enough continues, quietly, faithfully. You think of the simplicity within complexity. Although countless variables interact, photons, water molecules, ions, enzymes, stomata, leaf angles, and temperature fluctuations, they all converge to a single principle: sufficient, safe, sustainable action. Not more. Not less. Enough. And in that understanding, you sense comfort. The forest does not strive for excess. It thrives within bounds. Growth, production, and survival are measured, deliberate, and harmonious. Life respects the limits and flourishes within them. As the first stars appear in the night sky, you feel a gentle calm settle over the forest and over yourself. The lessons of light, water, enzymes, nutrients, geometry, and inefficiency all converge into one truth: the universe, in its quiet wisdom, balances potential with restraint and abundance with limitation. You drift slowly in thought, noticing the rhythm of the world. Photosynthesis may pause, but respiration and internal processes continue. Roots respire. Leaves breathe. Water circulates. Carbon cycles. Life persists within measured boundaries. Tomorrow the sun will rise again. Leaves will tilt, stomata will open, rubisco will catalyze, electrons will move, water will climb, and nutrients will flow. And yet the pattern will remain the same: the mathematics of enough, the quiet orchestration of limitation, the gentle rhythm of balance. You exhale softly and realize that this lesson extends beyond leaves, forests, and plants. Every living system, every ecosystem, every cycle is shaped by boundaries, by constraints, by limits that create space for stability, resilience, and longevity. And as night deepens and the forest rests, you carry with you the understanding that limits are not failures. They are harmony. They are quiet wisdom. They are the reason life endures. In the fading glow of twilight, you sense that the story of restraint, of light captured just enough, water used judiciously, nutrients allocated carefully, and energy managed with gentle imperfection continues silently, ready to greet the new day. And in that pause, you rest, comforted by the gentle mathematics of enough.